Well, so far you got the overall idea that it is really hard to pass the blood-brain barrier, and in this table you see the concentration of the drugs and some molecules such as glucose and the albumin as a protein within the plasma, and the amount of concentration which have been able to pass the blood-brain barrier. For example, in the matter of glucose, because the brain needs the energy, you see here that the difference between the concentration within the plasma and the concentration which has succeeded in passing the blood-brain barrier is not that much, okay? So they are close together. However, for some sort of molecules, such as the total proteins, as you see here, the difference is too much, okay? In the matter of, again, albumin, the difference is high. Even the immunoglobulins, they cannot easily pass towards the brain. And that's all you have to know about this table, okay? And you have to know that by increasing the molecular weight of the molecules within the blood vessels, their chance for passing the blood-brain barrier will decreases. Okay, within this slide, we just want to introduce how we can mimic, how we can simulate the tight junctions of the blood-brain barrier within an in vitro chip, okay, not in order to assess the permeability of some drugs. We cannot just give them into the animals and then kill them and cut the brain and into different slices and then see the concentration of the drug that could be able to pass the blood-brain barrier, okay? So by doing so, we want to mimic, okay? We want to create the same thing on an in vitro chip to assess the permeability. So here is the idea. We have the wells, you see here, these are the multi-well in vitro chips, okay? We have the well here, and in the inner well, as you see here, we culture the endothelial cells, and in the bottom well we culture the astrocytes and the pericytes and etc. Okay, and due to the presence of this liquid phase, they can contact with each other, okay, they can communicate with each other, and because they see each other together, the endothelial cells and the astrocytes, pericytes, they are stimulated to form the tight junctions all around here, okay? So we can solubilize a certain type of drug here and see by the passage of time, as the time goes by, how much of its concentration is able to pass through down here, okay? So we can somehow, not 100%, somehow mimic the tight junctions, okay? And by staining different parts of these cells which have been cultured, okay, the endothelial cells with the vimentin as a red fluorophore, and here you see the bottom part which have been stained with another fluorophores, and the occludin proteins with the green fluorophores. We will see the kinetics of the drug passage from the top layer to the down layer, and also the formation of the tight junctions due to the presence of these occludins and etc.